Hello, everyone, and welcome to today's webinar, Prep for Success, from Human Microbiome Exploration to Advanced SARS-CoV-2 Testing in Wastewater. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Dr. Sasha Vlassov, Senior Manager, Research and Development, Life Sciences Solutions Group, Thermo Fisher Scientific. Madhu Jasti, Staff Scientist, Sample Preparation at Thermo Fisher Scientific, and Emily Zeringer, Staff Scientist, Sample Preparation at Thermo Fisher Scientific. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Sasha, Madhu, and Emily, you may now begin your presentations.
Thank you. Hello, everyone. Today we'll be talking about the good and the bad bugs from human microbiome exploration to advanced SARS-CoV-2 research in wastewater and human samples. In our daily life, we primarily focus on macro objects. We observe human beings, animals, plants, other things, but there is an invisible universe of micro and nano objects. What this slide shows, not surprisingly, in terms of global biomass, plants are number one, and they're contributing the estimated 450 gigatons of carbon. Animals contribute only 2 gigatons, and the rest are microbes. Bacteria are at 70 gigatons of carbon, and protists, viruses, and other contributing more. So despite the small size, all these microbes are present at a combined huge mass, substantially more than all humans and animals on planet Earth. Most of the microbes populate the environment. They are found in a thick layer of soil and water, but many of them populate human body. Some of the microbes are our friends, some of them are our enemies, and today we'll be talking about both healthy human microbiome, some abnormalities, and deadly viruses such as SARS, which are coming from the surrounding environment and affect us, our health, and our microbiome. The human body, primarily the gut, is populated by 100 trillion bacteria and other members of the microbiota community, which play a fundamental role in our well-being. Bacteria are best studied members of human microbiome. Much more work needs to be done with characterization of fungi and bacteriophages. And it's intriguing to see that there are different submicrobiomes within the human body, such as skin, eye, nose, ear, mouth, etc. Different communities that populate parts of our body, and they're unique and different. Of the large number of functions and beneficial effects various microbes exert within the human body, only a fraction is well understood at the moment, because as you can imagine, the studies are extremely challenging. But it is truly impressive, even though this little segment that we understand is really amazing. For instance, it is known that human microbes prevent colonization by pathogens by competing for attachment sites and for essential nutrients. They antagonize competitors and foreign bacteria through the production of substances ranging from relatively non-specific fatty acids and peroxides to highly specific bacteriocins. Microbes perform carbohydrate fermentation and absorption, enabling the host to utilize some normally indigestible carbohydrates. They synthesize and excrete vitamins B and K in excess of their own needs, which can be absorbed as nutrients by their host. Microbes also provide a continuous and dynamic effect on the host gut and systemic immune systems. And finally, they also stimulate the development of certain tissues. It's a very impressive list, and this is just the beginning of the journey. Deviations from healthy microbial composition have been linked with many human diseases, including inflammatory bowel disease, obesity, cancer, asthma, diabetes, and allergies. There are many diagnostic tests and therapeutic solutions based on human microbiome, live bacteria, and the agents promoting or inhibiting growth of certain microbes. Some of these agents are in development, some of them are already commercially available. The most famous story is fecal microbiota transplant, or FMT, which was approved by FDA about a year ago as a treatment for recurrent C. difficile infection. This procedure is now used in every country in the world and every state within the United States of America. To summarize, we need to treat microbiome as a sophisticated system, much like the circulatory or immune systems that exist in harmony with homeostasis, play multiple roles within the human body, and the complexity is truly fascinating. There are different questions that can be asked by scientists and medical specialists in their studies of bacteria, viruses, and other microbes. Who is there? What can they do? What are they actually doing? And this will dictate the tools and workflows for analysis. Keep in mind that many bacteria, especially from our GI tract, cannot be cultured. And that's why only when molecular techniques, especially sequencing, became available, this area of research exploded. And it happened very recently, only about 10 years ago. So for the last decade, a tremendous amount of data was generated. Many exciting studies were published in the top-profile journals. And lately, if you attend different conferences, you can absorb plenty of information about this really exciting area of research. Today in our presentation, we'll focus on analysis of bacterial and viral RNA and DNA. We won't be discussing proteomics or metabolomics. These are separate areas of studies, and we'll be presenting on that later. Another topic that we'll be covering today is very exciting. It's SARS-CoV-2 research using human and environmental samples. The bulk of research and testing is done on nasopharyngeal swabs and saliva from different donors, but SARS is also reliably detected in human stool at the early stage of infection and for several subsequent weeks. Based on this finding, wastewater sewage surveillance became an extremely useful tool for monitoring the regional spread of SARS-CoV-2 and other viral and bacterial pathogens. Hundreds of organizations worldwide are successfully using wastewater testing for surveillance at the city level, as well as for zooming in as required for early catch cases, for instance, dorms at the university. Maybe many of you saw these announcements in the news over the last couple of months. Today, two of our scientists will be giving presentations. Madhu will first talk about the human microbiome research, tools and analysis by next-gen sequencing, and the qPCR and latest developments, briefly highlighting effects of diet and probiotics on the microbiome composition of GI tract. In the second part of the presentation, she will talk about the wastewater testing for SARS-CoV-2 surveillance. And then Emily will be giving a presentation about methods and workflows for analysis of viral, bacterial, and other pathogens by qPCR, highlighting the latest solutions for SARS-CoV-2 testing. At the discovery phase, when you don't know who is out there, next-gen sequencing is obviously the best approach, but once you know which organisms you want to detect and explore in detail, quantitative PCR using TaqMan Assays is the fastest, cost-effective, most sensitive, and specific approach. With that, I will pass the microphone to Madhu.
Thank you, Sasha. This is Madhu Jasti. I'm a staff scientist at Thermo Fisher Scientific. My presentation is about the human microbiome and gut health. What is microbiome and why do we need to know about this microbiome? Microbiome is a group of microbes in a particular environment. So human body consists of 20,000 human genes, and we also have 2 to 20 million microbial genes. That means for every one human gene, we have 100 microbial genes. And only 10% of our cells are human. We have 10 trillion human cells and 100 trillion microbial cells, 10 times higher. and the remaining 90% of the cells are from viral, bacterial, and fungal taxa. Over 100 trillion microbes exist in different parts of the human body. Out of these, the prominent microbiomes in the human body are in the mouth, which is saliva, has 20 billion microbes, and the skin has 500 species of microbes. That means for every square centimeter on our skin, we have 1 billion microbes. And the next one is the digestive tract, which is gut, which has 1,000 known species of bacteria, which contributes to 2 to 5 pounds of our weight. And out of all these prominent microbiomes, the most studied microbiome is the digestive tract, which is gut. Why the gut microbiome is more important? Because it is the largest, densest, and most diverse microbial community in the human body. There are several factors that affect the gut microbiota. That includes how the baby is born, like C-section or normal delivery, or how the baby is fed, formula fed or breastfed, and environment, genetics, lifestyle, diet, nutrition, probiotics, antibiotics, prebiotics, age, everything affects the gut microbiome. And every person has their own unique microbiome. It's like fingerprint. Every person's microbiome is unique. So how do we study this gut microbiome? Using the fecal DNA and RNA. Thermo Fisher offers a wide variety of tools for microbiome workflows. The first column here shows the tools that Thermo Fisher offers for sample collection and analysis of intact microbes. And the middle top portion shows the kits for microbiome analysis, Purling Microbiome Kit and MagMAX Microbiome Kit, which is the newly launched kit. And most of my data that I'm showing today is based on this MagMAX Microbiome Kit. And Thermo Fisher also offers the stabilizing solutions for nucleic acids like RNA later, and offers the instruments for automation, the KingFisher Flex and Duo. And for the downstream analysis of microbial DNA and RNA, Thermo Fisher offers the Ion Torrent platform and also Axiom Microbiome Array. Let's look at the data and the workflow for this MagMAX Microbiome Ultra Kit. MagMAX Microbiome workflow for rapid total nucleic acid purification. We have both options, manual and automated options for this microbiome kit. So basically, from the stool sample, it takes 50 minutes, less than an hour, to extract the total nucleic acid from the toughest sample like stool. So for the hands-on part in this workflow is the sample pre-processing. For this, we have two options, using the bead tubes and a bead plate. So we do the bead beading to lyse all the tough organisms, tough microbes in the stool sample. So for the high throughput option, we can do the 96 well plate. We can use the 96 well plate for 96 samples using the high throughput bead beater. After pre-processing the sample, the binding, washes, and elution of the total nucleic acid happens on the KingFisher. And if you look at the picture next to it, the red square shows how good the kit is. So basically, it's a high-yielding kit, giving yields up to 70 micrograms, and we can process 96 samples within an hour. It's the high-throughput option. And we have several protocols designed for several sample types using this kit. Let's look at the data, how the kit works. So the first picture here shows the 1% agarose gel with the total nucleic acid isolated from 6 donor fecal samples shown in duplicate. The first lane indicates the KD plus ladder shown for size comparison, and the top band is the DNA band isolated, and the bottom two bands are the RNA bands, the ribosomal bands for RNA, 16S and 23S. So this gel picture showing that the preps isolated, the total nucleic acid preps isolated using this kit are high quality preps. And to assess the RNA quality, the samples from these preps, RNA, is loaded onto the Agilent bioanalyzer using the RNA nanochip. And the Agilent bioanalyzer trace is shown here for the RNA. So basically, the RIN value, the RNA integrity number, is ranging from 8 to 9.5 for different donor preps. And you can see the quality of RNA on the trace here, showing 2 ribosomal bands, 16S, 23S, along with some small RNA. These results indicate that microbiome kit enables high-quality prep. Then we did the downstream analysis, the nucleic acid analysis downstream, using the applied biosystem TaqMan Assays. This is the TaqMan assay data for the DNA isolated using the microbiome kit, using the TaqMan probes for gram-negative bacteria, bacteroids, and gram-positive bacteria, firmicutes. So these firmicutes and bacteroids are the phyla, which includes several species in there. Basically, these probes detect multiple species in those classes. It's not like a single target. So the graph shown here, line graph, shows the CT values on the y-axis and the total nucleic acid extracted, analyzed from 6 fecal samples is shown on the x-axis. So we were able to detect good CT values for both the targets, gram-positive and gram-negative, indicating that this kit enables efficient isolation of total nucleic acid from gram-positive and gram-negative targets. And this is the data from the analysis of RNA using the same TaqMan probes for gram-positive negative targets, bacteroids, and firmicutes. And the CT is shown on the y-axis. So we were able to detect good CT values for all the 6 fecal samples, again indicating that microbiome kit enables efficient isolation of total nucleic acid from both gram-positive and gram-negative targets. So the next thing is, how does this kit work for other sample types? If you look at the vertebrate kingdom, we have several classifications there: mammals, birds, reptiles, and fish, and amphibians. So I was able to collect the fecal samples from all the vertebrates except the amphibians. So those were all tested along with some fungal targets. And the bottom picture shows the stool samples collected from all these different organisms. It's looking like a rainbow. This is the TaqMan assay data shown for all these samples collected and extracted using the MagMAX microbiome kit. The top graph is the 16S TaqMan assay data. So the CT values for the 16S, which is the microbe levels, are detected using this kit, using the several vertebrates here, like human, cat, dog, hamster, ferret. Those are the mammals and fish. And the reptiles included bearded dragon and emerald fish, and cockatiel is a pretty-looking bird. So from all these wide range of species, we were able to detect the microbiome, basically microbe levels with the good CT values. And the bottom graph shows the fungal target detection. So healthy donor stool samples were spiked in with the C. albicans and Aspergillus niger, which is the first 2 lanes shown there. And the last 3 lanes, these were collected from a grocery store, Red Star yeast and Fishman's yeast, and a probiotic gingerade. So from all these samples, TaqMan assay is done using the TaqMan probe for the total fungal target. So the CT values indicate that from all these samples, we can efficiently isolate the fungal target using this MagMAX microbiome kit. So these results show that this kit works for a wide range of sample types. So the next thing we wanted to see is what is the total microbiome profile. So this is shown with the microbiome case studies here. So to get the whole microbiome profile, we need to do the sequencing downstream, 16S sequencing, which is done on the Ion Torrent platform offered by Thermo Fisher. And just to give you some information before showing the microbiome profiles from each donor, so the most common bacterial phyla in the gut of healthy donors is mostly occupied by firmicutes and bacteroids, and to a certain extent, actinobacteria and proteobacteria. So let's look at the microbiome profiles from the total nucleic acid prep isolated with this kit. This is the case study 1. I wanted to see whether there is any effect of probiotics on the microbiome profile. So this case study is done on a single donor, tested for a single donor. That donor took 12 weeks probiotics, and stool sample was collected before taking the probiotics, before starting the probiotics, and every week of intake with the intake of probiotics. And the total nucleic acid preps were done using this kit and analyzed the bacterial profiles with the 16S sequencing on the Ion Torrent. So this is the data shown here. The heat map is generated by RStudio. The phyla level heat map is shown here, and the scale is given next to it. The scale basically shows if the scale is like orange to red in color, it's high level of those targets, and yellow shows the medium level, and the blue shows no or low levels of the target. So if you look at the before probiotics, the firmicute is at high level, and with the intake of probiotics by week 12, the firmicute levels are gone down. Bacteroids also started like before probiotic sample has medium level, whereas with the intake of probiotics, the levels have gone high. And coming to the proteobacteria, there is no significant difference with the intake of probiotics. For the actinobacteria, they started at the level before probiotics. They were like medium to low level, and with the intake of probiotics by every week, they have gone down. So this data is indicating that taking the probiotics changed the microbiome profile in terms of firmicutes and bacteroids for this particular donor. And the second case study that I did is effect of diet on the microbiome. So for this also, one donor was tested. That donor followed 16 weeks of diet suggested by a nutritionist. And this is a strictly vegetarian diet. And the stool sample was collected before starting the diet and after 16 weeks of diet. So the data shown here is the heat map generated by RStudio. So the bottom lane here shows the profile, microbiome profile from the sample collected before starting the diet. And the top lane here shows the sample collected after 16 weeks of diet. And the species detected were shown there, and the scale is given on the top. So the red squares shown here basically show significant differences in the levels of those bacterial species before diet versus after diet. For example, I can give you one example here. So the first one, allicite, that species is normally in high levels in a non-vegetarian eating people. So because this diet is strictly vegetarian, you can see that after diet, that species levels have gone down. Same thing goes with the other targets as well. So this result indicates that intake of specific dietary components was correlated with the microbiome profile for this donor. So the next topic is the wastewater surveillance. For this wastewater surveillance, why do we need this wastewater surveillance? It is basically needed for mass monitoring of SARS-CoV-2. So for mass monitoring, this SARS-CoV-2 is the biomarker. So basically, from the human body, human excretion, primarily the fecal stool, enters into the sewerage and transport via sewer. And we can take the samples from the sewage and test for SARS-CoV-2 levels to report the community-wide trends, or we can do the inter-community comparisons of the SARS-CoV-2 levels in that particular community. So to do this mass monitoring or wastewater surveillance, first we need to test whether we can be able to detect SARS-CoV-2 in the fecal samples or not, and we can be able to detect the SARS-CoV-2 in the wastewater samples or not. So the first sample tested for this purpose is fecal samples. So again, fecal samples in two different ways, raw fecal sample and the rectal swab. Both sample types were tested for SARS-CoV-2 using the spike-in of inactivated virus. So the workflow for the fecal samples is shown here. We take the fecal raw sample or the rectal swab sample and add the lysis buffer, add it to the bead beating plate, do the bead beating, and collect the supernatant after the bead beating, and take the supernatant for the total nucleic acid extraction using the KingFisher instrument. So for this workflow, I did the 4 spike-in amounts for the SARS-CoV-2 for all the raw samples and the rectal swab samples. And for the raw fecal samples, I have tested 10 donors here. And for the rectal swab samples, 12 donors were tested here. So this is the data for the fecal sample testing with the SARS-CoV-2 spike-in. So this is the TaqMan assay data using the TaqMan probes for SARS-CoV-2 specific genes, M gene, ORF1-AD gene, and S gene. And also did the inhibitors test using the Xeno. Xeno is an internal positive control offered by Thermo Fisher to assess for inhibitors presence in the total nucleic acid during the extraction. Since stool samples have high amount of inhibitors, this Xeno test is shown to show that whether the samples have inhibitors or not. So the TaqMan assay data, the first graph is for the N gene, shows that the CT values are shown on the y-axis, and all the 10 fecal donor samples extracted is shown on the x-axis. The 4 spike-in volumes here, the 1,000 copies, 500 copies, 50 copies, and 10 copies of SARS-CoV-2 is spiked into the healthy donor stool samples here and extracted the total nucleic acid. So the N gene data shows that for all these 10 donors, all the 4 spike-ins were detected with good CT values. For all the N gene, ORF gene, S gene, we were able to detect all these 4 spike-ins. And the Xeno graph shows that we detect the same CT for all the 10 donors comparing to Xeno control, showing that if there are any inhibitors in the total nucleic acid extracted, we expect a CT shift. But there is no CT shift, indicating that there is no inhibition from the extracted total nucleic acid. So this result is showing that we can detect as low as 10 copies from these fecal samples with these spike-ins. And this data is from the rectal swab samples for SARS-CoV-2 spike-ins. Here again, 4 different SARS-CoV-2 spike-ins were done, 2,000 copies, 1,000 copies, 200 copies, and 20 copies. And TaqMan assay probes were used for N gene, ORF gene, and S gene. And Xeno assay is also done to check for inhibition. And as you can see here also, like before, for the rectal swab samples also, all the 4 spike-ins were detected in all the 12 rectal swab samples extracted here. Same is the case with the S gene and ORF gene. And the Xeno graph also shows no CT shift from all these 12 donors extracted compared to Xeno control, indicating that no inhibition from the extracted total nucleic acid from the wastewater, from the fecal samples, rectal swab samples. So the next sample type is the wastewater samples. So these wastewater samples, we got these from 4 different counties, and this is the picture showing how the wastewater samples look like. And we have used 2 methods of pre-processing, direct method and a spin filtration method. Before explaining how we proceeded with that pre-processing, we collected some analytical parameters for these wastewater samples, comparing to the nucleus-free water. So pH, conductivity, oxidation reduction potential, and the ion selective electrode, which is basically the ion concentration in all these wastewater samples, comparing to nucleus-free water is shown here. So pH is same for all of them, including the clean water, and conductivity is high for the wastewater samples compared to the clean water, and the oxidation reduction potential, that is within the normal range for all the samples, including the clean water, but except wastewater sample 3 has in the negative range, indicating that that sample has more reducing substances in the wastewater sample. And the ISG, ion selective electrode, ion concentration, is high in the all wastewater samples compared to the clean water. So basically, these analytical parameters are needed to design how to treat the wastewater. So the next thing is, how did we pre-process these wastewater samples? The first method we followed is the direct method. So we take the wastewater samples directly into the multiple wells in a bead beating plate, do the bead beating, cool the supernatant from the multiple wells, and add 2 ml of the supernatant per well in a 24 deep well plate, and do the total nucleic acid extraction on the KingFisher and the downstream analysis with the TaqMan Assays. And the second method of pre-processing the wastewater samples is the spin filtration method, which is, so we spin down, we take an aliquot of the wastewater sample, spin down at 10,000 G for 30 minutes to clear all the large debris, and we take that supernatant and filter through the Nalgene 0.45 micron filters, and take that filter, chop that filter out, and chop those pieces, add those pieces into the bead beating plate. And once the bead beating is done in presence of lysis buffer, those supernatants were taken and total nucleic acid is extracted, and downstream analysis is done on the qPCR instruments using the TaqMan Assays. Let's look at the data for these wastewater samples. So this is basically without any spiking of SARS-CoV-2. We wanted to see whether the samples that we received from these counties, whether they have any detectable levels of SARS-CoV-2 or not. So for this, we process them same way shown before, spin filtration and direct method both. So the graphs here show the green dot shows the spin filtration method, and the black dot shows the direct method of processing. And a negative control included here is the PBS, phosphate buffered saline. So the TaqMan assay data here shows that PBS, no CT detected for PBS here, if you look at the N gene graph. And for all the wastewater samples, they have detectable levels of SARS-CoV-2. And this is the same case with the other genes as well, R and S genes. So this data is indicating that viral load is detected even without any spike-in. Next, we process the wastewater samples with the spike-in of inactivated SARS-CoV-2. So this is the data with the direct method of pre-processing. So TaqMan assay data for N gene, R gene, S gene, and the xeno data for inhibition assessment. So if you look at the N gene graph, all the 4 wastewater samples spiked in with the 8,000 copies and 4,000 copies of SARS-CoV-2 were giving the CT values around 23 to 24. And same is the case with the R and S genes. And the xeno data also shows that there is no CT shift comparing to xeno control, indicating that the total nucleic acid extracted from these wastewater samples are free of inhibition. So this data is indicating that viral load is detected with the spike-in of the inactivated SARS-CoV-2 in this direct method. And the second method of pre-processing, spin filtration method, this is the data processed by this method, and same thing as shown before. So here the spike-in amounts used were 5,000 and 2,500 copies. So the CT values were detected for both the copies, spiked in for N gene, S gene, R gene. And the xeno assay also shows no inhibition, no CT shift comparing to xeno control. So to conclude, MagMAX Microbiome Ultra Kit is for faster purification of inhibitor-free microbial DNA and RNA from challenging samples. And we have the automation-ready protocol designed for KingFisher Flex and Duo systems. And this is the fast procedure, allows for 96 samples processed within an hour. This is the high-throughput option. And this kit is compatible with a wide range of sample types, such as stool, wastewater, and swabs contained in the transport medium. Along with these, I have shown you all other vertebrates and some crazy samples also. And this kit enables reproducible recovery of high-quality nucleic acid compatible with broad range of applications, including microarrays, qPCR, and next-generation sequencing. And I would like to thank my team, my mentor, especially Sasha, with a poop looking like statue there, and my wonderful teammates. And my suggestion is eat healthy, stay healthy, and balance your microbiome. Thank you so much for your time. And Emily, you can take over.
Thanks, Madhu. Thanks to all of our attendees for dialing in today. My name is Emily Zeringer, and today, for this last part of our webinar, we'll be talking about pathogens and microbiota, detection, monitoring with molecular methods, kind of a bit broader range organisms used for molecular methods. So first, quickly on the agenda, I did want to go a brief introduction behind molecular methods and kind of their benefit and how kind of we look at them. And a few more details around the workflow around molecular methods, including the benefits of using high-throughput protocols and instrumentation as part of the workflow, and then case studies are examples of real-world samples with these high-throughput methods. So first, the introduction. So when we think of microbial and pathogen studies, we kind of look at them as falling into one of three main areas. The first area being viral detection, specific viral detection, and this can include RNA viruses, DNA viruses, single-stranded, double-stranded, positive, negative. These type of workflows also include, can also include easy-to-lyse bacteria. The second area we focus on for studies would be, we call it the all-pathogen detection. So this will include the viruses and gram-negative, easy-to-lyse bacteria, but will also include the harder-to-lyse organisms, such as the gram-positive bacteria, as well as yeast and fungi. The third main area is for microbiome research, which Madhu provided a very detailed and comprehensive description of. And so in the following slides, I will focus more on the viral detection and the all-pathogen detection. However, for all three areas, it does encompass multiple sample types, ranging from biofluids to more solid types, such as fecal samples, as well as sputum. And so for each of these areas, for the viral and then for the all pathogen, detection of microbes and pathogens is nothing new. It's been around for quite a while. The more traditional methods, kind of the gold standard, they work very well, but they are limited in certain ways. They are low throughput and often have a narrow focus, looking for one analyte or one target. And often you require multiple workflows to get the same answer, answer in different ways from the same sample. And so you can see it kind of laid out. You have your sample type, your micropathogen of interest, your extraction method, analysis method, and each step often has multiple options or multiple protocols that must be completed. So in this day and age where, the number of samples is increasing and the amount of time desired to get answers from these samples is reduced, these traditional methods, aren't as useful. And so kind of to lay it out in a more visual schematic. So here you can see you have two samples, sample A, sample B. You're not unknown microbiota in each of these samples. And for each one, you have to follow this workflow. You perform your purification, in this case, DNA RNA purification. If you're looking for virus, bacteria, and fungi, these could potentially be 3 separate workflows you're looking at. And then for each one, is it PCR? Is it immunology, culture? So you can imagine for each sample, it can take a few hours. They are very quick tests to a week or more, especially if you have to culture. And so for 2 samples, not so bad. But as you start to increase the number of samples, the amount of testing increases almost exponentially. And so it becomes kind of a burden as far as time and effort. And so one of the large benefits of the molecular workflow is it takes this, what can become a very complicated system and simplifies it. And so you end up with a very simple and comprehensive workflow for pathogen detection. It also allows very targeted analysis of pathogens. And so you're able to look at, you have a list of organisms of interest, and you're able to look at those very specifically. So if you look, so again, you have your sample type A, your sample type B, but both of them go through the same workflow. They go through the DNA RNA purification or total nucleic acid purification, followed by quantitative PCR. And so you're able to start from your sample, get to your results in a very manageable amount of time. And again, so you have the same workflow, and so you increase the number of samples. However, your workflow doesn't change. You're still able to have your very simple and comprehensive protocol or workflow, even though you've increased your samples, in this case going from 2 to 8. And so not only can you look at specific samples, but it allows for kind of more of a screening monitoring of a larger number of samples as well as a larger number of microbes and pathogens. And so that's kind of the basic introduction to the molecular workflow, but then a little bit more details about the workflow. And so when I say workflow, this is essentially what I'm describing. It looks, laid out like this, it is relatively simple until you start getting into the details. It gets more complicated. But the first thing, as with most, you start off with sample collection and storage. We won't really delve deep into this today. We just don't have the time. But this can be very critical to good results downstream. So once you have your sample collected and then stored or shipped to you, then it goes through the sample lysis. Really what I'll be focusing on in this part of the webinar is the enzymatic or chemical lysis. I think Madhu did a really good job describing the mechanical bead-beating lysis used for the microbiome kit. And then once you've lysed your sample, you go into actually extracting nucleic acids and then taking that nucleic acid downstream into real-time PCR. And so these 2 areas, your sample lysis, nucleic acid extraction, and downstream PCR, you have quite a number of options as far as methods in order to do this, all the way from low throughput to high throughput or more automated workflows. And so if you look at them separately, for sample purification, you can think of it as having three flavors. You have your spin columns, your organic extraction, and then your more high throughput or automated system. And so spin columns and organic extraction, they're going to be more of your traditional methods, organic extraction, trizol, or similar spin columns, such as a pure-link column. These are the gold standards and work very well. However, they tend to be more labor-intensive, require more hazardous chemicals, and often can require centrifugation. So for a smaller number of samples, they work fine. But if you're starting to get in the tens, the hundreds, even the thousands, it becomes very untenable to use these two methods. And so that's when you would switch to your automated system, which can vary in types. For us, we use a magnetic bead-based system, which can be performed manually, or as we like to do it with instrumentation to relieve the burden of hands-on time. And though they work very well, very useful for high throughput, they are not without their limits. Everything really is a trade-off. You get your higher throughput, but you are, the protocols are more fixed, your application areas are more fixed. But it does offer a good option for pathogen detection. In addition, you do also have throughput options related to your real-time PCR, all the way from your single-tube assays, which are your most low throughput, ideal for a fewer number of samples, and does, I guess you could say, offer the most flexible option as far as number of assays to be run. And then you have your arrays. These either come in a plate or card format. The plates allow for a larger number of samples for a fewer number of assays, whereas the arrays, a fewer number of samples, a larger number of assays. These would fall more into your low to medium throughput options. Also, they come as fixed, but they also do come customizable. And then you have your final offering, the most high throughput option, and these are your open array plates. Like the arrays, they are pre-spotted, either with the standard offering or a fixed offering, or customizable, and allows you to process up to 48 samples, and then even up to a larger number of assays. And so you can imagine for one run, you're able to generate a very large amount of information. These open array plates are probably most useful for screening or profiling type of experiments. And so I've kind of spoken briefly about the high throughput options for the workflow, and now I kind of want to go a little deeper in kind of what we offer and then the benefit. So really the benefits, there are multiple benefits. I will say probably the two biggest ones we talk about are going to be user time and then consistency. And so user time or hands-on time is another way to think of it, is a huge time suck for a lab. If you have to have a tech performing the extraction and it takes a full hour of hands-on time, that's a lot of time. And then you add a number of samples and that can increase exponentially. And so by using a high throughput or even an automated workflow, you're able to, the same amount of time, the extraction may take the same amount of time, but the actual hands-on time is 5 or 10 minutes, leaving, if you have an hour protocol, 50 minutes that the tech is able to do other tasks. And so your user time becomes more productive and less focused on the task. You can do more multitasking. The other is inconsistent results. Anytime you have a step which requires human hands, the potential for variability is much higher, as opposed to using a instrument or automated system. Having a robot do it, you'll get much higher reproducibility versus, or much less variability versus human hands. And so those are the two biggest areas that high throughput methods or protocols in the context of molecular pathogen detection workflows that they can benefit. Three other ways. The potential for increased sensitivity. Again, you're using a system that has more automation or more, instrumentation, which allows increased consistency, which can help with sensitivity. Already mentioned the reduction in user hands-on time, but again, significant time savings because from one sample, say up to 96 samples, it's the same amount of time. And so look at that as more bang for your buck for the same amount of time. And then, of course, the increased productivity. If you don't have a tech sitting performing an extraction for an hour, if they're only hands-on for 5 minutes, then, that's more they can get done in a day, and also potentially more samples that can get run in a day. So, high throughput, and especially high throughput combined with automation, really can provide a very useful and productive workflow. And so for us, I mentioned that for our automation, our high throughput, we use a magnetic bead-based extraction system. We also combine that with instrumentation, specifically our KingFisher instruments. And these run the gamut from automation light using our Duo Prime system, which has the benefits of an instrument to perform most of the extraction tasks, but it's essentially a low throughput. You can only run 6 to 12 samples. But, to trade off a small footprint, smaller number of samples, but you get the benefits of the consistency. And if you're only running, say, 20 samples, 24 samples a day, that system works great. The next on the list is our semi-automation offering. So this is going to be our KingFisher Flex system. The nice thing about this system is you can run up to 96 samples or 24 if you're using the deep well plate. But it provides a medium to high throughput method where you get the benefit of automation and automated system for consistency, but you do still have some upfront steps as far as liquid handling or filling. And then finally, we have the full automation option, which is our KingFisher Presto, which really, to get the full benefit of this system, you have to combine it with the robotic liquid handler, something like a TaqMan or a Hamilton. And this provides you your walk-away option, as well as an ultra-high throughput option. Again, similar to the Flex, 96 to 24 samples per run, but still, you're able to walk away, which reduces the hands-on time even further. And so with these instrument systems, we've also developed chemistries to go along with them to provide users with the easiest and most comprehensive options. And so I'll focus on really 2 of our pathogen detection kits. So the first is our MagMAX VirallPathogen nucleic acid kit, which should be very familiar to most people by now, especially if you're working with SARS-CoV-2. But this kit covers several slides ago I mentioned the 2 areas we focus on for microbial and pathogen detection. So this is going to cover the viral detection, as well as some gram-negative easy-to-lyse bacteria detection. What's nice about it is it's pretty flexible. It's compatible with a large number of samples, large number of viruses, flexible volume input range, but the extraction time is relatively short. That's 25 minutes, and that is 25 minutes on the KingFisher Flex instrument, for example. And you can see data we've shown for multiple sample types. Blood, VTM is just viral transport media, and urine for multiple different viruses as well as gram-negative bacteria, indicating the ability of the system to work across multiple organisms, sample types, and provide a very flexible option. Really, our second offering, this covers the all-pathogen focus area. And so this is MagMAX VirallPathogen Ultra Kit. And so it also covers the virus and gram-negative bacteria, but it does include the harder-to-lyse organisms such as gram-positive, candida, yeast species, as well as fungi. Like the MagMAX VirallPathogen, it covers a wide range of sample types as well as a wide range of organisms. Extraction time is longer in this case, but that is because it includes an upfront enzymatic lysis, really to be able to extract nucleic acid from the gram-positive and the fungi or yeast species. And so these are used most often for this data here, especially is extracted using our KingFisher Flex instrument. However, it is also compatible using our Presto system combined with a liquid handler. In this case, it was a Hamilton Nimbus system. And so here you can see just comparing with the Flex to the Nimbus, results are equivalent for two different input types and for a range of targets. And so again, just showing the flexibility and benefits of a high throughput system, or automated system, however you need to use it. So now I'll move on to the case studies. And all of these case studies use our high throughput automated systems, both for the extraction as well as the real-time PCR. So the first is looking at the MagMAX VirallPathogen Ultra kit. Here we had a respiratory tract microbiota open array plate. It targets 44 different pathogens, and so we wanted to be able to see what we could detect from extraction from bronchoalveolar lavage samples or ball samples. And so using those samples, we used them straight or combined them with different spike-in organisms, extracted with our MagMAX VirallPathogen Ultra system and KingFisher Flex, and then performed the open array protocol, real-time PCR protocol, which includes a pre-amplification step. The entire workflow, we can process up to 96 samples within 8 hours. And so a very relatively fast system, which gives you a very large amount of data to work with. And so so here outlined, so these are all the different targets included on the open array plate. And then from our spike-ins, these diamonds indicate the expected positive samples for the spike-in positives lavage samples. What's interesting is looking at all of the samples, we knew what we should detect, and then so we were able to detect all of those. And then we were able to confirm samples that had previous test results, and then also able to detect a large amount of samples that we didn't know were in, large amount of organisms that we didn't know were in those samples. So, a very useful system and high sensitivity. And so here is just kind of outlining. The green indicates what should have been detected based on our spike-ins. So you can see the spike-in samples detected them, whereas the no spike-in samples did not, so that looks good. The purple indicates what we did detect, but were not spiked in, so potentially present in those different samples, which is very interesting. And then the blue indicates results, matching results from previous detection. And so all that was detected previously was redetected when we tested these samples again. So able to generate a large amount of data, relatively short amount of time, and a low amount of effort. The same is true. We did also have an open array plate, which has vaginal microbiota targets on it, and so using the same protocol, we're able to kind of have similar output, just a different list of organisms. Also with our MagMAX VirallPathogen kit, just looking at virus and gram-negative, we have done comparisons against kits out in the field just to confirm that we were performing as expected or as customers would expect. So here we're looking at negative single-stranded RNA virus, and then positive single-strand RNA virus, and then double-stranded DNA virus. So compared to the competitors, equivalent or actually improved performance. And then we were interested to see how low could we go as far as viral copies into extraction. And so we were able to find for human blood and human plasma detect as low as 50 copies spiked into a 400 microliter extraction for CMV and EBV. Kind of more relevant to what's going on in the world today, the MagMAX VirallPathogen kit, as well as its later version, the MagMAX VirallPathogen 2 kit, have been used to extract SARS-CoV-2 virus and then detect using TaqMan Assays. And so you can see we were able to correctly detect 3 of the gene targets for SARS-CoV-2, as well as a extraction control for 2 different volumes at different concentrations. And so actually a pretty sensitive system that works really well. And then finally, our most recent extraction protocol, the interest in saliva, or the ability to use saliva as a sample type for viral nucleic acid detection has skyrocketed, especially over the last couple of months. And so we wanted to make sure that we had a protocol that would match or be able to be used with saliva. Due to the nature of saliva, it does require its own protocol. It cannot use the same protocol as, for example, a viral transport media. And so we developed a protocol for the MVP and MVP2 kit for raw or stabilized saliva. And so here you can see one of our tests is we took saliva either raw and diluted in PBS or from stabilized and gene fix reagent, spiked in some inactivated respiratory pathogens, and then extracted them using the MVP1 and MVP2, and then detection from real-time PCR. So here you can see we use the 5 viral targets are from the spike ends, and then 16S as an endogenous control. And so what you can see for over 48 saliva samples between the raw and the stabilized, you see equivalent results for the 5 spiked-in virus, as you would expect. And then 16S was detected in all samples, but at different levels, again, as you would expect. And so, using a high-throughput automated methods provides you, for both extraction and for PCR, provides a very comprehensive workflow, flexible workflow to reduce hands-on time and get results quicker. And so that was all I had. Thank you all very much for your time, and I'll pass it back to the moderator for questions.
Thank you, Sasha, Madhu, and Emily, for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question goes to Emily. If we want to extract nucleic acid from gram-positive bacteria and fungi in saliva, which kit would you recommend?
For this, I would recommend the MagMAX VirallPathogen Ultra kit. It has the upfront enzymatic treatment, which will maximize recovery of nucleic acids from the gram-positive bacteria and fungi.
Okay, thank you, Emily. My next question here is for Madhu. Most of the microbiome studies are focusing on human or mouse stool samples. What was the most exotic sample type you tested?
I have tested like wide range of sample types, as my presentation also shows several vertebrates and birds also tested here. And the crazy samples that I tested is from the grocery store, yeast samples, and also like the dust collected from the vacuum cleaner. So those were all tested. And also the most important sample type tested is the parasites. So the gut stool sample has so many parasites if they have IBS syndrome or some C. difficile infection. So we were able to detect the parasites. We were able to extract the parasites with high efficiency with this kit.
Thank you, Madhu. My next question is for Emily. What are the differences between the saliva protocol for the MagMAX VirallPathogen kit versus the original protocol for use with other sample types?
So the two protocols are actually pretty different. They do follow the same basic outline of sample pre-processing, lysis, extraction, elution, but one of the biggest differences was the pre-processing. We actually, in the protocol, have very specific instructions on how to treat the saliva sample, both raw and stabilized, before going into the actual extraction process, mainly because saliva samples are so variable between collections and between different donors. The pre-processing steps we developed help to kind of normalize that, so it increases your chance of a good extraction.
Thank you, Emily. My next question is for Madhu. Can we use KingFisher Duo for automation of MagMAX Microbiome kit? What are the pros and cons versus KingFisher Flex?
We can use the Duo for the MagMAX Microbiome kit. It was already tested and it gives the same yield as the Flex. So the source is basically we can use less consumables because if the customer has few samples to process, they can go with the Duo. If they have more than 12 samples to process, they can use the Flex instrument to use the 96 deep well plate. And also, like the yields are similar, as I already mentioned. The only cons for the Duo is we cannot process more than 12 samples at a time.
Okay, thank you so much, Madhu. My next question is for Emily. What is the average time to process 96 samples using the high-throughput extraction, high-throughput PCR open array workflow?
So average time, it does depend kind of on what open array plate you're doing, but I would say the average time is about 8 hours, and that's from start to finish. That's from you have your sample in hand to you have your results off the instrument.
Okay, thanks, Emily. My next question is for Madhu. This is our last question. For SARS wastewater surveillance, you showed concentration of the water samples using filters. What other concentration options do you recommend?
There are like wide variety of options and included in the publications like worldwide. Like some people do PEG precipitation and some people do ultracentrifugation, but we didn't test all these options with our MagMAX Microbiome kit. But when time permits, we wanted to test the PEG precipitation on these wastewater samples. It seems like it's going to concentrate the wastewater samples pretty well because we have tested with the cell culture media and it works pretty good. So we need to test more options on this.
Thank you again, Sasha, Madhu, and Emily, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. The next installment of the Prep for Success webinar series takes place February 11th and focuses on vaccine development, so please join us for that. Until next time, goodbye.